I'll start by giving like a brief, the outline's going to be like a brief, like my scientific trajectory. Then I'm going to be talking about a little bit of history of tissue engineering, possible lines of research, differences between like the different methodologies that we have in tissue engineering, and some of the stuff that we've been doing at the lab, as well as current and new collaborations. And then I'll just wrap up with, well, I had some learning objectives. I think you all saw it. They were sent to you, but yeah. So with no further ado, so these three people, and the reason why I always start my talks with these three people, this is because when I was doing my clinical rotations at Mass General Hospital, these were the three people that actually made me change my career into research. And the reason why is because I was all set in mind that I was going to do cranial malformations as a specialty in plastics. And I ended up at the burn unit just because there was a fire incident in Rhode Island in 2003. And when I got to rotate in the Department of Plastic Surgery, it happened that these patients were in the OR, they were just flown in and they were at the burn unit. And we all had to put all our efforts into, getting these patients out up and running, right? So they were happy when they left the hospital because they were alive. For me, the outcome was horrifying, right? And I started questioning myself why we haven't done anything in all these years, all these centuries. The outcome looked exactly the same as Egyptians when they used to like just dress them all up and then have them get their wounds healed by their own. And why we didn't do anything. So that led me to pursue a PhD, and I came here to BC to do a PhD in plastics, and specifically in burn and wound healing. And I started studying cell-cell communication. I'm not going to go into the details of my PhD, but I'm just going to go in some of the details of the things that I did while I was doing my PhD. So I got to work with Steve Boyce. He's a professor of Department of Surgery in the University of Cincinnati, and he's one of the pioneers for cultured skin substitute. So what you're seeing here in your slide is how he was producing the skin substitute. And actually, he was able to do a clinical trial in Edmonton at the burn unit with Dr. Tredget. And the patient that you're seeing right there in the like below is a patient that was seen at the burn unit in Alberta, in Edmonton. And those are the skin substitutes that were placed. And as you can see, well, the outcome after 479 days post-op or post-dressed, they're not as bad as the previous patients that you saw. But if you see the edges where the skin substitute is grafted or where we put the skin substitute, still there's a hypertrophic scar. And why is this happening? So also another thing that I realized, and I'm going to just go back a little bit. Well, yeah, so that why is this happening? So we wanted to have, or one of the things that I wanted to do was have a skin that was readily available for patients. Because when you have 80% of your total surface burned, like there's not a lot of donor sites that you can get skin from. And so why not having like a skin that was already there, like almost like shelf ready, and you can just place it in the OR. And so one of the things that I did, this was obviously at the research level, was working with the skin substitute that Dr. Boyce created in a rabbit ear. So the rabbit ear is it's a specific model for hypertrophic scars. And so what we did is we transfected fibroblast with IDO. So IDO is an enzyme that is expressed in trophoblast cells and Sertoli cells, and basically in cancer cells as well. But basically what it does is it creates a physical barrier for the immune cells. And so it doesn't let the immune cells actually to penetrate. And that's why, for example, we don't reject the fetus when we're pregnant. And so it has that kind of immunity level, right? So we transfected fibroblasts, which they don't express normally like IDO. We transfected one with IDO. They started expressing IDO, and we placed the graft and the rabbit ear. Now, what we saw over time is that the scar was improved, and there was a well-organized, cells were well-organized in the matrix. But most importantly, you would say like, well, so what about like the T cells? So this is what I was talking about. So when we have the skin substitute with IDO, you see how like you're in this, I mean, obviously it doesn't make a lot of like justice to it, but you can see, do I have a mouse? you can see how the T cells are mostly aligned where the graft finishes. Now, one could say, like, how do you know that the graft is right there? Well, we also saw like vascularity, like in the dermis part. So it also favors that. But how do I know that the graft was still in place? Well, I did an HLA staining because I did place fibroblasts that were human fibroblasts, and rabbits don't have that HLA. It's not the same. So when I did that, well, I see that the graft is still in place after 35 days. So without any rejection, right? And the ones that I just put the graft without the IDO, the one in the middle, then there's no graft anymore. Nothing is staining for that graft. So with that, still there's problems, and the problem mainly is that when you're in the OR and you're trying to place a sheet that looks like a piece of paper, contouring that sheet to the irregularities of the wound and also the topography of the wound was difficult. So a colleague of mine who was also doing a PhD, we were sitting at a bar one day, and over beers we were like, I said, like, wouldn't it be fascinating if we had something that we could just pour into the patient and solidifies in the patient, and then it will like all form like the contouring of the wound. It will fill out the topography, also like the irregularity of the wound, and it would be much more easier to work with. And so this person, who is now my colleague and partner for a company, he is the one that his entire PhD was to develop this. So he developed a liquid skin substitute that is thermosensitive. And what it means basically is that it's liquid when you apply it, and it solidifies when it when it reaches 37 degrees, and that's when it's in contact with the body surface, or with the body itself, not the surface. And it's easy to work with. The cells, I mean, they love it. I mean, we haven't had there's a ton of papers, actually 4 papers. I just put 2 in there, but there's 4 papers backing up like this product. And now, well, it got patented through UBC, and we got the license out of UBC, and now we started this new adventure, which is connecting. Oh, sorry. It was just. So then I moved on, and I went to do my
Is that in clinical trials now?
I had a clinical trial already in Iran, and right now the idea is to, have that here. But it's not that right now. We're in the funding stage, so, but yes, that's the next step. And it's actually patented as a medical device because it doesn't, you don't have to have the cells in it, so it doesn't require theoretically a trial. But anyway. So then I went on to I continued my adventures at Northwestern. And so I went on to my postdoctoral fellowship at Northwestern University in Chicago. That's when I met David many years ago. And so one of the things that, so I still continued working on skin and how to regenerate skin. And so one of the things is like, well, and I'll talk about that, but 3D bioprinting, which was also big at that time and still is, was out there, but we couldn't really replicate the vascularity of, the complex vascularity of the skin or other organs. And so I thought, well, why don't we take advantage of the human body, and we use that vascularity to start growing, in this case, skin. And so I imagined a in vivo bioreactor. And so this is the first sketch that I did, basically. How did I imagine this thing, which obviously it's not like this, but I imagine it like a kind of a modular device that you can place on the patient. The patient would be in there, and then you can put like a decellularized scaffold, and I'll talk about that, but you can put any scaffold as a material, and you can perfuse through an automated system. You can perfuse nutrients, you can perfuse cells, and you can start growing, that, in this case, skin. And so that's basically what I envisioned. I wrote down what components would it have the device, if we wanted to do muscle, because burns it's not only for skin, but when you have a deep burn, it could also affect the muscle. It can injure the muscle. It could go all the way down to bone. So all these different elements, we have to regenerate them as well. It's not just putting a layer of skin. So I envisioned all these components, and then, well, I did some experiments that I'm not placing here. I mean, this is how we did it in animals. So we had a little chamber sitting on their backs. and as the in vivo bioreactor, and this would be hooked to the computer. I don't have a picture with the computer, but you see those inlets. So those would be the ports where we would hook it to the computer and then perfuse cells and perfuse nutrients. And we had a matrix inside. So all that trying to grow this skin. Now, again, not going into details, but there was a lot of data and work that it was done. And then we ended up patenting this. And, so I did show the data actually. So this is the decellularized matrix that we had inside. And this is the tissue after extracting it from that bioreactor. So as you can see, we threw in some cells. We threw in actually adipose-derived stem cells, but I'll get into that. And we start seeing vascular formation here. And we didn't get any infection. And then I'll show you also when we put it in the bioreactor in a further slide, how we also get like hair follicles and stuff like that. But for now, this is what happened. And we patented this, and then the patent got sold to the US Army. So this was part of a consortium where Anthony Atala was also part of it. But then, as you know, when you're trying to regenerate or engineer any organ, is what kind of cells are you going to use? Differentiated cells sometimes are not an option since either, as I told you, you don't have donor sites, or the cells have already a disease, or they're already they're basically not available. So we need to start looking for alternative sources of cells. And so I mean, again, plastics, you get a lot of fat, and the fat tissue is usually discarded in the OR. And a matter of fact is fat tissue actually yields 5 times more stem cells than bone marrow. And instead of having like a bone marrow, like getting a piece of bone marrow and getting some of those cells, why don't we use like the fat that is usually discarded in the OR? And so we started, so that's why I gave a talk in terms with the title was From Fat to Skin, because here was my idea was, well, we can isolate and purify adipose-derived stem cells, which I did, and then we sorted them. This is just to show that actually they are adipose-derived stem cells, because then we transform them into, or we differentiate it into adipocytes, which is on your 7 days. That's just a confirmation. But what you can do with these adipose-derived stem cells is that you can actually co-culture them with the cells that you want them to become. And apparently, not only they change morphology, as I'm showing here in a co-culture system, but they also change their genes and the gene expression and the protein expression. So here what I'm showing is that I started seeing markers, specific markers from keratinocytes expressed in those adipose-derived stem cells. So we call them keratinocyte-like cells, because I can't say that they're keratinocytes itself. They're like cells because they seem to behave exactly like the keratinocytes. So they were expressing all these genes, and they were expressing, the proteins were also expressed, which is more important than actually the genes. And then I said, well, I need to prove that there is a function to this. So one of the functions of these cells is that they stratify. And so creating this barrier that we know. So then I made them, I just placed them on a decellularized dermis. And I start seeing that when I was culturing with EGF or with the keratinocyte condition media, they will start forming the stratification, as you can see right on this side. So this is normal skin. This is how it looks like, and this is how it stains for the different markers. And this on your far right side is the adipose-derived stem cells, or the keratinocyte-like cells that were behaving very similar to on the other panel. And when I just throw adipose-derived stem cells with no stimulation, no signals, these don't do anything. Basically, they actually got into the matrix, and likely, I didn't test for this, but likely they would just become fibroblasts because they're mesoderm, like it's the same from the same lineage. So we did that. Then, as I said, we decided to say, well, if I use adipose-derived stem cells and I put them in the in vivo bioreactor and I put the appropriate factors, like stimulating factors, what happens? And well, as you can see, if I add the appropriate growth factors, I start seeing this structures that look like hair follicles. And if I don't throw the growth factors, then nothing happens. Like even like these, the adipose derived stem cells were GFP labeled. So you can see that they're still here, still forming some things. They must have like some signaling also from the in vivo, but not there yet and not as clear as you see in the other one. So then, I mean, still in Northwestern, I wrote another grant for functional muscle regeneration, and very similar, using our in vivo bioreactor and with a piezo motor, you can have stretching, and you could, the idea was to create an environment where you can regenerate muscle. So muscle was decellularized, was implanted into animals, and then we have this stretching part. And then for arteries and veins, well, we had a graft, right? So we did an AV loop, and then we had a nerve grafting as well. And this, when I left, was still ongoing. So I just left the lab with this grant for 5 years, and that money that you see in the bottom. But really, so what is tissue engineering? Well, for now, I think you already understand that, tissue engineering is merging that biological methodologies with the engineering methodologies to create tissues that can be later, that we can replace from the damaged ones. And so this was coined again back in 1987 when the term was coined. But really looking at the history, we've been talking about tissue engineering since almost ever. There are Sanskrit there's Sanskrit findings where they talk about a skin grafting. And so skin grafting probably was, according to that, it was done at that time, right? And then we have many other things where people, like, for example, that painting, that it's historical painting, where Comas and Damien, actually, they were replacing legs, right, from wounded soldiers from one to another, right? And again, either was it just myth or just, still, people were already thinking about these things, right? But again, as I said, it wasn't until, this term was coined. And then from there, it was a little bit of a boom coming out. And then again, it stopped because of several issues. And I'll talk about that. Like, FDA stopped approving the use of stem cells. Then there was the fiasco of Paolo Macchiarini, that now I think it's available on Netflix. So all of you could actually watch it. But tissue engineering actually had it was hurdled because of this. But anyway, so we have the skin probably was dermis was probably one of the first ones to come out. Alloderm was commercially available since before the 2000s. And then we had these pulmonary valves also available. This is a cryovalve, like Singer, from a company as well, that it's available. And these are all decellularized tissues. Then we had also printed tissues like bone tissue, cartilage tissue, all these coming up. But again, when this was set up, Langer and Vicanti said, tissue engineering needs at least 3 components, right? And these are the 3 pillars. So cells, the scaffolds, growth factors, those are the 3. And I'm not going to go into details, but you can imagine that for cells, all like stem cells, you can refer to stem cells to differentiated cells. Scaffolds, you can think about it like natural, synthetic, decellularized ones that I'll talk more in depth about that. And growth factors, all of them. However, only PDGF is the only one that is FDA approved. So we talk about all these growth factors, but we can't really use them in patients, right? Because the clinical trials have failed. Again, we can discuss about that, but I think it would be another entire topic. So again, going into those methods that I've been talking about. So the most current ones, or the most trendy ones, I would say, is 3D bioprinting, as you have heard, and decellularization. Now, 3D bioprinting started actually at the end of the 1990s. So I think it was 1997 when Holt actually, thought about, can we use a machine that, has cartridges and, you can put ink in there, and, everything from the computer or from a computer can be translated into a piece of paper in a 2D fashion. And then from there, we came up with a 3D bioprinter, which actually, can we print in 3D, in a 3D structure, in a 3D manner, tissues or other things, right? Like stereolithography was one of the first ones. And then now, the idea is that we can eventually one day print tissues that we can use for our patients. So instead of using those cartridges with ink, filled with ink, we can fill them with extracellular matrix proteins. We can fill them with cells, right? And, having different heads and combined, we can start printing organs. Obviously, this is a far-fetched, like heart is not there yet. None of the solid organs are there yet, but the principle is that you would deposit, the combination of extracellular matrix protein with cells, and you will start applying it like, layer by layer by layer until you grow whatever structure you want. And for that, all you need is a CAD design or the MRI information. Like you can put it into the computer, all that kind, like CT scans as well. They work with the software as well. And then eventually have an organ like this. Again, still not there yet, but we will someday, I hope. So the other method that I wanted to talk about is deserialization, right? So deserialization is, instead of reinventing the wheel that it's trying to do with the 3D bioprinting, is why don't we use already the organ? So the organ, basically we remove all the cells from the organ and we leave the extracellular matrix. And for that, well, we use a combination of methodologies like physical, chemical, or enzymatic. And it's never one, or it's usually two, the combination of two of them. It sometimes can take all the combination of three of them. And so these are some of the, now, how do we consider, or how do we decide that we have successfully decellularized an organ? Well, there are two things in general. One of them is having a total cell removal, and that we do it with looking at the double-stranded DNA. So we quantify that, and if it's less than 2% of that total DNA, and that's per milligram of tissue, then we can consider that the decellularization was done effectively. The other thing is the preservation of the microarchitecture, and this is the most important thing, because this is the reason why we're doing decellularization. Because as you can see in the top panels, this is a liver lobe, and when we decellularize it and we inject through the vessels, we inject some kind of polymers. Oh, sorry. We can see that this is what's left behind. So this is from a native one, and this is from a decellularized one, and you can see how it's very similar. So we really preserve, we're really able to preserve that capillarity, which is what it's very difficult to replicate using a 3D bioprinter. So, but each of them have limitations, as everything, right? And so for organ decellularization, still we're working on the cell receiving techniques. We haven't been able to really completely rebuild the parenchyma of some of the organs. We have to use sophisticated bioreactors, and that could be costly. For the 3D bioprinter, the 3D bioprinter hasn't been able to really print complex vasculature and very complex structures, especially because we're talking about sometimes nano or micro scale. And so that is not, that hasn't been the resolution of the printers. The other thing is the size of the organs or the tissues that we want to print. Like this is based on the head, or not the head, but the plate where we print these things, or the table that we print these things. And that has obviously limitations, because then you will have to have like a 3D bioprinter that is way bigger in order to get bigger organs. And the other thing, and this is the most important thing that I want you to pay attention to, is the combination of the extracellular matrix proteins. And I say this because we can do proteomics of the matrix of an organ and know exactly what's the composition of the extracellular matrix. But the problem is that, and this is the example that I give to students, is like, well, we know that buildings are made of concrete, they have like some wiring, they have, glass, they have piping. But if I mix all that together and I throw it in the middle of the street, it's not going to be a building. And that's exactly what we're doing when we're trying to 3D bioprint. Now, yes, the cells will eventually remodel all that and create a new structure. But the decellularized organ already has that in place. Okay? So the decellularized organ is basically, we throw a bomb in the middle of the city, but without destruction of the buildings. We just, destroy all the people in there. And what it's left is basically all the buildings with water running, everything functioning. And now we bring a new population of people to the city. well, first, even if they don't know the city, they'll start wandering around and saying, well, this looks like a building for working, and this looks like a building for, for living, and, and this looks like, oh, this is tap water. Oh, we can drink this water. So these are the kind of examples I get, and this is how I see it. So this is how I visualize it. But still, sorry, stem cells and growth factors still not approved by FDA, so that's a big limitation for both of the technologies. Now, possibilities, well, the sky really is the limit because we can use it for research applications, we can use it for clinical applications. So really from 3D models that we're using in the research stage, right, where we want to study how the interactions of cells, cell-cell interaction or cell extracellular matrix interaction, like drug testing, for example, personalized medicine, things like that, right? And in the clinical use, obviously, like we want to develop this to grow tissues and organs ready for transplantation. So some of the things that we've been doing at the lab. Well, so in 2018, I was successfully awarded with a CHR grant that I wrote, and this was the first one. I remember talking to Alan and saying, like, we should write a CHR grant, and he's like, oh no, that's big leagues. Like, don't get disappointed if you don't get it. And so actually the first one I wrote in 2017 and was proposing exactly what you're seeing right there, which was a 3D patient-derived tumor model for personalized drug testing. And that was merging basically my expertise with Alan's expertise of, kidney cancer or bladder cancer. And so with that, I tagged along with Dr. Black and Alex Wyatt, and Alex Wyatt for the genetic validation. We'll talk about that. And Dr. Black, because of the expertise as well in getting the clinical samples from bladder cancer patients. So the whole purpose or the whole idea was, well, can we get tumor samples, right, from kidney, well, from bladder in this case. That was a grant proposed specifically for bladder, but the idea is any cancer. You get samples from the patient, you dissociate this, and you get the cells, right, from this tumor, and then you seed those decellularized organs or decellularized matrices from that specific organ with the cells from that patient. And the idea was to start drug testing and knowing which one is the most efficacious drug to give to the patient. So with that, again, we started decellularizing bladders, pig bladders in this case. We proved that the decellularization was successful. So as you can see on the top right, you see the double-stranded DNA quantification, which is now going almost to zero. And we did also scanning electron microscopy, and you can see how no cells were left behind with H&E also in scanning electron microscopy. And then, well, Cesar, and this is all the work from our Ph.D. student that is, his name is Cesar, but you probably haven't known him. And so Cesar, the first thing that we did is from those tissues, from those decellularized scaffolds, I said, well, first start, kind of optimizing your technique and use UC3 cells as a trial instead of wasting our patient samples, right? So he started inoculating or seeding these cells into the decellularized matrices, and he treated them with cisplatin and gemcitabine. And this is what he saw. Well, he saw a decrease in the proliferation, looking at Ka67, and an increase in tunnel assay. So seeing that as he was increasing the dose of cisgem. So that was his first trial. But then once he had, and this is just, I'm showing 2 slides, but really is the work of lots of months and years, right? Doing and trying to optimize this technique. But then once he was steady and and everything was good, we use now the sample. So at that time we had collected, I don't know, it was close to probably 30 specimens. But the problem was that because we don't do it retrospectively like most of other studies, we do it prospectively. So some of those, when the pathology report came back, they weren't muscle invasive or even bladder cancer. Some of them was Irwin sarcoma that we had somewhere there. And, we had tissues from, or it wasn't a primary, it was a secondary. So those we had to discard a bunch. And we ended up with 17 samples that were bladder cancer at that time. So from those, we did genetic validation, and that was all done in collaboration, as I said before, with Dr. Wyatt. And unfortunately, from those 17 patients that we saw, some of them were non-muscle invasive. So we had to discard that. So we ended up with 13 samples that were muscle invasive. And from those, all the 13 samples were done. Cesar actually did the 3D model that he seated it in the decellularized bladders that we had. And so he had those, and he treated them with Cisgem to see what was going to be the drug response in those tissues. And that we compared it to the clinical outcome. Now, unfortunately, when we went back to look at the files, like the charts from these patients, some of them decided not to get any drugs. They didn't want it to go for treatment. Some of them were lost to follow up. And we were left with 7 samples. And those 7 samples, actually, there was a good correlation between the outcome from the patient and the outcome from our model. So that gave Cesar a reliability of 71.5%, although now he has increased that number because more patients have come up and we have a better outcome. Now, you would say like, well, this needs to be compared to other models. What are the existing models? So then I collaborated, I created a collaboration with Dr. Karen Chung in the biomedical engineering school. And she is big on working with microfluidics. And she created this way of making spheroids with a microfluidic, using a microfluidic chamber. And so we started making these spheroids from the patients that we had. And this is just a video of how the setting was in the lab and in engineering. And so Cesar compared the spheroid model with the 2D culture model, which is the conventional way of, looking at drugs and doing these tests. And what he ended up seeing with the same 7 patients that we had, and what he ended up seeing is that our model still had more reliability than the spheroids and the 2D model. So moving on, but still my vision, we can use it for research as I mentioned, but still my vision is to create tissues and organs that are ready available for transplantation. And the way I see it is that everybody is a donor. If I die today, people can retrieve my organs, all of them, and then these organs can be decellularized and kept in biobanks and keep them there ready for whoever needs them. So if I know that there's a patient, or not me because I will be dead, but if somebody knows that there's a patient with end-stage renal disease, for example, and we're at that stage yet, then we would take out an organ, a kidney, from those biobanks. We can, match it with the size, not with HLA or anything like that, just might match it by size because obviously a pediatric patient will require different size of kidney, but we can match it by size and start reseeding it with the cells from the patient. Now, you will ask which cells from the patient because they're going to be already, the fenestrated ones are already diseased. Well, so this is where I'm going to be talking next because it's going to be stem cells, right? We have to think about stem cells because there's no other option for us. So we will be reseeding it and then transplanting it back to the patient. And then this transplant is going to be an isograft. It's not going to be an allograft anymore and likely is not going to need immunosuppression. So that's basically my vision and the vision of other people that are seeing this as well. So for that, we had another student, Anna, which she was great actually, and she just recently graduated. But her thing was to start growing kidneys in the lab. And so for that, before her, we were doing it in rats and mice. So we were decellularizing kidney, rats, and mice. But we really had to upscale it because, that doesn't cut like the bill. Like it's not the same. If we want to get ready for the future, which is transplantation, we wanted to upscale it to a human size organ. And so that we went into pigs. And we didn't buy pigs for just the sake of killing them and getting their organs. We basically tagged along with other researchers or other projects or even like the trauma clinic that they use pigs all the time in our animal facility just for clinical training purposes. So we got the kidneys. We would get 2 kidneys from the pig. One kidney, we would actually dissociate that and get all the cells from that kidney. And the other kidney was decellularized. And the reason why we were getting the cells also is because first we wanted to do proof of principle, optimize all our procedures. And for that, we needed kidney cells. So easy just to get the kidney cells from the pig as well, and then reconfigure it again or engineer it again in the research part. So this is basically a flow diagram of what happened. And so again, we got the kidney, we would dissociate the kidney, and then we would just get the cells from the kidney, that as the first stage, culture those cells. And we also looked at what kind of cells we were looking at. Also, we wanted to know what kind of passage was ideal to use these cells. So as you can see, it actually looks pretty small, sorry about that. But we went all the way to passage 4. And we start seeing that in the early passages, yes, there is a switch, for example. So we start getting more stroma cells, and we start losing some of the important cells like aquaporins, CD31, and podocin, for example. So what we decided is that we definitely need to use cells at an early passage. And this is just for, again, optimizing our protocols. As I told you, like the future is obviously we'll be using stem cells, so it has nothing to do with this. But this is just to optimize what flow are we going to do, what kind of rates are we going to do, how many cells do we need to perfuse, et cetera, et cetera. So we did immunohistochemistry, and we stained these for the different types of cells. So we see that we do have we do get a lot of different types of cells. Although I know that the kidney has 26 different types of cells, but here we tested for the ones that we could get the antibodies for. And so then we went on to obviously the decellularization, and you can see, well, when we decellularize, again, we're very careful. So the matrix is, we really preserve the microarchitecture. So as you can see, this is a native kidney. We see beautiful, like the Bowman capsule, and we see the glomerulus, and then, or the glomeruli. No, it's actually glomerulus because it's only one. But anyway, and then when we move into the decellularized part, we still see that preservation of the Bowman capsule, and we see this, it's collapsed because there's no cells. But once we start perfusing with cells, and also the technique of how it's done, like the SEM, but the loops are still there. So we did proteomics to the extracellular matrix because we wanted to know, well, what are those components? And so we compared that to the native kidney. And so in the native kidney, we found 4,000. This was with the help of Hans Adamet. So the native kidney, we see 4,610 proteins that we got hits for, and the decellularized one, 3,164. So there's roughly like 1,500 proteins that we don't see, and this is because there's no cell components. So most of those proteins of the 1,500 that we don't see are pretty much, or must be, like cell proteins. But these, we get the extracellular matrix. And interestingly enough, we've seen that before, but also other researchers have reported this, we still get like growth factors. So growth factors are still like attached to that extracellular matrix, and likely, and this has been reported not by us, but reported by other people, likely this is what is driving that migration of stem cells, and also making them differentiate into the different, like the types of cells of that particular organ where you throw it in. So again, this needs obviously more exploration, but these have been some of the suggestions. So we still get these growth factors. Now, going back into our diagram, if you remember, so we already got the cells, we already decellularized the organ. Now it's time to cellularize this organ. So we started recellularizing, and again, I'm just showing a snapshot of one slide, but this took a couple of years for Anna to get this. Actually, no, like a year and a half. And this is only 12 days, and I'm not saying that we will be able to regenerate an organ in 12 days by any means, but this is the time point that we decided to stop when we saw this. And so this is 12 days of this kidney. Now, you see it's congested, and I'll tell you why. Well, because when this is at 5 days, this is at 5 days. It has no blood in there. Well, no erythrocytes, actually. I wouldn't say like we actually perfuse with blood, but it has nothing. So we were just oxygenating these cells by oxygenating the media. But that's not enough. Cells, we were constantly monitoring for LDH. We were constantly monitoring for other, for different things. Also, for other key markers that they will tell us that cells were starting to die. We were looking constantly at the media that was the output basically of the media, and we were looking if there were cells in there, if the cells were dead, what was going on in that media. So we started noticing that we really needed to oxygenate this, and in order to oxygenate this, well, what better than an erythrocyte, right? That's basically their job. So we would draw human blood, and we just got the erythrocytes. We extracted all the other components of the blood, and we just perfused or we mixed the media with the erythrocytes to make them as an oxygen carrier. So we were oxygenating now the media with the erythrocytes, and those were transporting the oxygen into the kidney. So that's why you see it almost purple. Because I think I don't think, we actually added too many erythrocytes, but it was our first trial. So this is what when we section that kidney, this is what we see. Lots of erythrocytes, as I say. But we start seeing this component, see? So if you look at this, if you take a close look, you start seeing this round structures here. We also see that where there was a vessel, we would see it, that there were cells around the vessel, right? And so this is another one comparing like native decellularized and recellularized and how it looks like. So again, this is our, one of the, not first attempt, but after 7, 8, or 9 iterations that Anna did. This was the last one before she left. And well, we do see a better progress, especially better outcomes that what other people have reported in the previous experiments that is recorded in the literature. And then we wanted to see, well, what kind of cells are we getting there? And so we stained the tissue for podocin like CD31 by Ment and aquaporin and aquaporin 4. And we start seeing that the round structures, solid round structures that we were looking at, they actually do look like, I mean, they were podocin positive. Then you have like these tubules that were formed, they were CD31 positive. We had like stroma cells like there also present, aquaporin 1, for example, and aquaporin 4s like right here, right, forming these structures. So definitely cells started migrating, and they're migrating to the right places. Again, we're not there yet, but it's just, you know, a start. And we looked at LDH quantification just to see if there was any suffering or any cells were dying. And, you know, I mean, yes, by day 9 all the way to day 12, we didn't see a significant change. But really, like the levels of LDH units per liter, calculated units per liter, were way less, were way below what normal serum levels show. So from there, also we've been doing, again, Cesar has been working on decellularization and recellularization of pig bladders. Again, this is in working progress, so I don't have like right now slides to show you. But again, looking at the cell source, at this alternative cell source, adipose-derived stem cells, since I have already the expertise, I've done it for keratinocytes. I said, well, why don't we try this again for kidney cells or for even like bladder cells? And so I know pretty well Dr. Carr and Dr. Papp, and I said, well, can you give us like fat tissue from the clinic, and we can start isolating these adipose-derived stem cells. And so obviously there's a human ethics protocol in place, not that it was just under the water, but with that, we started isolating these cells and then looking at their protein profile, genetic profile, and functional assay. That's eventually where we're going to get into. So we isolated the cells again using flow cytometry, and we do get the pure or almost a pure population of adipose-derived stem cells, and Cesar co-culturing them with SV Hawks, which are the normal for epithelial bladder cells. He starts getting changes in morphology, very similar to the SV Hawks. Not all of them change in morphology. As you can see, there's still some cells that don't look like SV Hawks, but others that do. And what he saw is that as he was increasing passages, these cells were becoming more and more uroepithelial-like cells. So he looked at the genes, at the gene expression, and he saw that they started expressing, for example, this is cytokeratin 5, whereas adipose-derived stem cells don't express cytokeratin 5. Cytokeratin 7 was kind of odd because we don't see any significance, and it seems that adipose-derived express it, and there's some papers out there saying that at the genetic level, but when we look at the protein level, it's really not expressed. So that's good because then no matter, we know that sometimes genes are expressed or upregulated, sorry, not expressed, but they're upregulated, but the protein not necessarily is expressed. Yeah, I'm done. And well, these are the controls where we see that adipose-derived stem cells still have like their CD90 and CD105, which are are specific from adipose-derived stem cells that the urothelial-like cells and the SB don't express. The same was done for kidney. So for kidney cells, we do see change in morphology, and we start seeing the expression of aquaporin 1. This is still in the works, so the students are still doing this work. And I mean, and then just moving into the collaborations, and I'm going to go very quickly. So we started a collaboration some months ago with Dr. Ryan Flanagan and the deserialization of human testes as a model for studying male infertility. So we got testes from a human patient, and we decellularized that testis. This is what it looks like. And then we have looked at that, the decellularization was done well. And the idea is that we also harvested some cells from that tissue, from that testis tissue from that patient. So we have the cells, and the idea is to repopulate that. So this is actually a work in progress as well. And Megan, her research associate, actually does have another sample of the decellularized one because they're going to be lyophilizing that and putting it into the 3D bioprinter. Other collaborations, again with Dr. Ryan Flanagan and Horacio Bach, we're doing the development of a smart matrix for bone regeneration. So again, this is preliminary work that it's been done. So this is how we're doing nanoparticles, and we're encapsulating that. So we're trying to encapsulate growth factors that are osteoblasts like to migrate and to proliferate, and so we're encapsulating that and making this as a gradient in a matrix. So we call this a smart matrix because it will allure basically the regeneration of bone. But that's also a work in progress. We wrote a CIHR last year, but we withdraw it just because we didn't have enough preliminary data. So we're planning to send it for this next competition in the spring. Also, I have this collaboration with Dr. Jelena Jančić at the University of Duquesne. And she's doing synthetic oxygen carriers because, as I showed you, like the erythrocytes is not the solution for oxygenating the organs because the erythrocytes take space. So it's better to use a oxygen carrier. So she's doing perfluorocarbons, and she has already published some of them for prolonging the solutions that to preserve kidneys between retrieving it and to transplanting it. So using the same, that she will have to tweak it, but using the same principle, we will be using it to oxygenate the organ. And then future collaborations. Well, yesterday I had a meeting with Dr. Dirk Lang, and then we were thinking about providing an in vitro 3D model for studying kidney microbiome. I spoke before with Dr. Forbes and maybe decellularizing the prostate to develop 3D model to study BPH. And I do have a standing collaboration right now with Dr. Vijay Barantla, which is decellularizing free flaps for reconstructive medicine, and that's at Wake Forest. Also, like through the industry, like I proposed a budget and it got approved for $450 per year for 2 years in the development of engineering fat tissue. So how does this really fit into the plan of this Institute of Urological Science as well? So at least I see a clear fit with 3 of the different centers or departments that they're going to be aligning here. So one of them, we already have one with the sexual health, androgen, and infertility with Dr. Ryan Flanagan, kidney transplantation and immunology, obviously, because, well, that's my whole interest is to create tissues for transplantation, and obviously with the prostate center as well, and the onco part and the oncology part, creating 3D models. So really, I leave this to you guys, but is tissue engineering the future for urology? So, I mean, I hope that one day we get to the far end, that we can actually print in patients, like directly into patients. We can use them, as the left side shows you, as for research purposes, like to do personalized medicine, like to look at the best drug for that patient. But really, like, I see the future. Let's see if everybody sees the future. So with that, this would not be possible, obviously, without collaborators, students, but most importantly, without the that support that Dr. Allen so gave me from the beginning and letting me pursue my dreams. So thank you very much.